I'd like to begin by welcoming everyone here. I'm Nancy Maisels, and I'm the director of the University of Washington Molecular Medicine program. This is a program that was designed to capitalize on the recent progress in research at the interface of basic science and medicine, and a program that capitalizes on the amazing advances that have been recently made in developing new diagnostic procedures and in developing new therapies for human disease. And at the University of Washington, we decided that all of this excitement meant we had to do something new in PhD training and bring some more elements of medical training to our graduate students. So we developed this very interdisciplinary and interdepartmental program, which is a collaborative effort among the Department of Medicine and the basic science departments of biochemistry, bioengineering, genome sciences, immunology, microbiology, pathology, pharmacology, and physiology and biophysics. And they've been very supportive of this program, and we're also fortunate to have received support from the Howard Hughes Medical Institute, which is partially funding this lecture tonight. So one goal of our program has been to convey our excitement to people in the University of Washington community, the Seattle community, and to anybody who's able to watch a lecture on UW television. So we decided to start this lecture series, and this is the third of 5 lectures we've had this academic year. We're very, very lucky tonight to have as our lecturer Dr. Jerry Nepom, who is the director of the Benaroya Research Institute at Virginia Mason. And Jerry has really devoted his entire career to diabetes and to ameliorating the effects of this disease on human patients. I got to know Jerry a long time ago when he was an undergraduate at Harvard, and I was there too. And Jerry then went on to enter the MD PhD program here at the University of Washington. He went back to Harvard. He was on the faculty there for a while. Fortunately, the Hutch recruited him back to Seattle, and since 1985, we've been lucky to have him here as the director of the Benaroya Research Institute, which has grown enormously and wonderfully under his direction. So I'm totally delighted to introduce Jerry Nepom. And Jerry, thank you for coming to lecture tonight.
Thank you very much, Nancy, and thank you all. It's a pleasure to be here. I'm going to be discussing with you tonight research involved in predicting and preventing autoimmune diabetes. And I'm going to start by defining with you what we mean by autoimmune diabetes. It's a term that some of you may not be familiar with. You've all heard of things like juvenile diabetes and type 1 diabetes. Those are synonyms for autoimmune diabetes. Autoimmune describes the situation when the body's immune system attacks itself by mistake. We all think of our immune systems as something that protects us from viruses, protects us from bacteria. That's true, but the immune system makes mistakes. And when it attacks our own bodies by mistake, that's autoimmune. Now, diabetes, of course, is the disease when the sugar in the blood gets too high. That results in the disease called diabetes. And so putting the two together very simply is autoimmunity is the cause and diabetes is the effect, giving us type 1 or juvenile or autoimmune diabetes. Now this is what it looks like if you were to look at diabetes under the microscope. The cells in the body that make insulin are found in the pancreas, in these little mini organs called islets. You'll learn a little bit more about islets in a few minutes. And this slide shows you a picture of the islets under a microscope on the top panel there labeled normal. Those cells are the cells that are making insulin. Insulin is the hormone that regulates the level of the sugar in the blood. You need insulin to live. because without it, the blood sugar goes too high, causing fatal diabetes. The bottom panel shows you what the same islet, the same type of piece of the pancreas looks like in patients with autoimmune diabetes. What you're looking at are all these little black dots, and each one of those black dots is an immune cell that's from your immune system, circulates in the body, and is out there to protect you from all kinds of infections and things. The immune cells have made their way into these islets in the pancreas, and they're in the process of killing the cells that are making the insulin. So that's the difference. The top panel is the normal islet making insulin. The bottom panel is the islet that's been infiltrated with immune cells, and those immune cells are killing the cells that make insulin. So fundamentally, why does this happen? It happens because, and this is the bad news, I hate to be the one to break it to you, but our immune system is very error-prone. Okay? It's a part of the nature of the immune system, the way the immune system is developed and how it adapts to different pathogens and things. The immune system is fundamentally error-prone. And when it makes mistakes, those mistakes can cause autoimmunity, can cause autoimmune diabetes in this case. But what is it fundamentally that underlies the error-proneness of the immune system? Well, it comes down to a variety of things. Some inherited genes, we'll talk about that in just a minute. Infections that can trigger it. Other environmental exposures, including chemicals, toxins, and drugs that can influence the immune system. And ultimately, sometimes just random molecular events that can happen by chance are part of the triggering mechanisms. Okay. Now we'll talk about some of these in some detail. And the way I'd like to describe it and sort of lay out the format for this discussion is as a pathway here, taking us along the disease course of the immune system when the immune system makes these mistakes, when it runs amok, as it were. And the immune system takes us through 3 different discrete stages before we actually get to diabetes. There's a stage of susceptibility when the immune system starts to go wrong and creates in our bodies a situation where we're susceptible to the autoimmune attack. That stage is followed by a second stage when the immune system's actually activated, actually starts to turn on, begins to start the attack on the tissues that make insulin. And that step is followed by a third step where there's actually destruction of the cells that are making the insulin. And that's actually the final cause of the clinical disease. And one of the reasons for thinking of it as a stepwise progression is because it matters. It matters to the patient. It matters in terms of designing therapy. And it matters for diagnosis. So for instance, if we can determine this first step, the susceptibility step, that helps us understand who's at the highest risk of diabetes in the population. If we can understand the second step, how does the immune system get activated to attack the islets, that helps us figure out how we can stop it and how we can design clinical trials to try to intervene. And the third, when we can measure this, that's the time to jump in with major immune therapies to try and treat the disease. So we'll go through these 3 steps a bit in the course of this talk. First of all, let's start with the susceptibility. I've already warned you that some of the susceptibility is because of genes that we inherit. What's illustrated here is that there really are just a small set of genes that make most of the difference. There are actually a lot of genes that we have that modulate the risk a little bit for or against the disease, but there fundamentally are only a few that really make a big difference. The HLA genes here are the major players. Depending on the specific combination of HLA genes that you inherit from your parents, you can have an increased risk of diabetes up to 30-fold compared to people who don't have those particular genes. Some of these other genes we'll mention briefly confer substantial but much lower risk, around twofold, compared to people who don't have those genes. So a combination of different genes tell us who in the population is at the highest risk. What do you need to know about those genes? You don't really need to know their names, but these are the points that sort of matter. First of all, these genes are very common. In the Pacific Northwest here, close to 20 to 24% of the population carry one or more of those risk genes. Okay? But the good news is that they only confer risk. They don't cause the disease. And that's because of the sequence that I've already introduced to you, that there are stages of the disease. So the genes give you the risk, but they don't give you the disease. Okay? What do the genes do? We'll talk about this in just a minute because they do something very important. They control the way the immune response knows to attack the cells that make insulin. That's fundamentally what those genes are all about. The bad news is those genes are actually also sometimes useful for normal immune responses. And why is it bad news? Why did I call it bad news? That's because once we figure out that the genes are responsible for targeting the immune response on the cells that make insulin, a logical therapy would just be to get rid of them, get rid of those immune cells based on targeting the products of those genes. But we can't quite just do that because the same genes are useful in different contexts for some of our normal immune responses, and we don't want to do anything bad about that. So it gets to be a little bit tricky. Now, what those genes are all about, in terms of the specificity that I'm talking about, how do they know to target the cells that make insulin? I've diagrammed it here schematically for this discussion. On the left is one of the immune cells. You remember that original picture I showed you of all those black dots sitting on the islet there, infiltrating the islet? I said those were immune cells. Each one of those black dots is an immune lymphocyte. This is a cartoon of an immune lymphocyte showing you some molecules on its surface. This cell circulates around in the immune system and sees a variety of other tissue cells that are called antigen-presenting cells. You don't need to know the name. Just the point is that there are receptor recognition systems that are required to signal the immune lymphocyte to turn on, to become activated. Okay? That signaling goes something like this. They just physically come together and trigger the cell to activate. It's very straightforward. Now, I've used a color code on this slide because the red indicates the 4 components that are made by those 4 genes that I just told you about that are the susceptibility genes. So the answer to the question of what do the risk genes do is the risk genes make these molecules that function in this kind of immune recognition. Okay? And these are the 4. This big one right here we'll come back to a little bit later. That's the HLA. That's the product of the HLA gene. It's fundamental to this kind of intermolecular communication. So when that happens, when those gene products are there and when they get connected like this, you get the second step that we talked about earlier. The second step, immune activation. So this diagram shows us our immune cells here, these little green cells with their molecules on the surface, searching around, looking for the islet cells that they're specific for. When they find them and when they attack them, that's how you start the process leading to type 1 or autoimmune diabetes. It's when the immune cells get activated. And we call that pre-diabetes because, as I told you before, this is only the second stage. The cells that make insulin are not yet gone. The immune system has just been activated against them. What should you know about this second stage, about the immune activation? First of all, these immune cells are specific for insulin-producing cells. That's why people with type 1 diabetes or autoimmune diabetes, by and large, don't have other autoimmune diseases. Sometimes they do, and when they do, it's because the immune cells see additional targets around the body, like targets in the thyroid gland for autoimmune thyroiditis, or targets in the adrenal gland for a disease called Addison's disease. But most patients with type 1 diabetes have immune cells circulating that are very specific for targeting the cells that make insulin. What do the immune cells do? They initiate an immune response against the islets. And what that means, the implications of that, are that once you initiate an immune response, it's like calling in all the other troops that are around. The immune response is very diverse, lots of different kinds of cells. Some of you may be familiar with some of the other cell types, B cells, NK cells, cytolytic T cells. They all get recruited in, cause a very active inflammatory process, cell damage process. That's important, and the reason I stress it here, because at this stage, in the second stage, some of the therapeutics that we're now trying in clinical trials are targeted at the original cell, the original activation event, and some of the therapeutics that we're trying are targeted at the other elements of the immune response that get recruited in to cause to propagate the damage at this state. And the third point here is also very important. One of the things that happens at this stage, at this activation stage, is that different immune cells talk to each other. The T lymphocytes, the subset that has this receptor system I've been talking about, talks to another subset called a B cell subset, and trigger the production of autoantibodies from those B lymphocytes. That's the main clinical importance of that, is that the autoantibodies can easily be measured in the blood of anybody, of patients or people who are potentially at risk. And they serve as a biomarker for this immune activation event. So now I've told you two things that can be measured that take somebody off the street and identifies whether or not they have gone through this first stage of susceptibility and the second stage of activation. Those two things are the genes and the autoantibodies that can be measured. And so we, along with large consortia in this country, funded by the National Institute of Health, have developed very large screening programs that have screened over 200,000 people so far for these genes and for these autoantibodies to identify who's at risk and who is progressing down this pathway. There may even be people in this audience that have participated in these trials. I see a couple people nodding already, actually. So you know that from a very personal point of view that how this is done. And it's a way to identify who does not have diabetes, but who is either at risk if they've gone through the first stage, or who is both at risk and has had some immune activation if they have both the genes and the autoantibody positivity. Okay. Now, that takes us to our third step, which is the actual destruction of the cells that make insulin in the pancreas, the damage step. Now, this step is fairly complex. It takes a long time. Most of us have, when we're born, about 1,000 clusters of insulin-producing cells called the islets. And each islet contains up to about 1,000 cells that make insulin. So you have, when you're born, you have close to 1 million cells that make insulin. And the process of damaging them, and so they lose the ability to make insulin and cause diabetes, that process takes time. Sometimes it takes months. It can take years. I've just schematically illustrated it here as a time course from birth to along a flat line to the point at which the damage is initiated, and then progressively islets get lost and the curve starts to go down until there are no more surviving islets. Okay? I'm going to show you on the next slide an enlarged version of this graph with a lot more detail. As I've already told you, when you're born, we can identify the genetic predisposition, but you go along the flat part of the curve here without loss of any beta cells until there's that immune activation event. I've already told you there are multiple triggers for that, and I'm not going to have a whole lot more to say about that because we're going to mostly be talking about this progressive decline of the curve. as the curve goes down, meaning that you're losing islet cells, progressively losing the cells that make insulin. And what's important about this curve is that initially, even as you're losing islet cells from the immune damage, you still can have normal insulin release. Our bodies are remarkable. We're born with more islets than we need. So you can lose a few and still have normal physiology. But once you've lost a bunch, and a bunch means 50% or 60%, you start to enter a high-risk zone where the body's ability to make enough insulin isn't quite there. And at the point where you've lost about 3/4 of your islets, that's the point where you start to develop symptoms and you go to see a doc and you're diagnosed with type 1 diabetes, overt diabetes. Okay, so this is what the time course looks like. And one of the objectives of a lot of current research is to be able to measure this and monitor this so that we know when a person who's at risk and has had some immune activation. So we know when a person has started progressing down this slope and is a good candidate for intervention therapy that we're going to talk about. Now let me populate this diagram with a few numbers so that you have some idea about the scope of what we're talking about. I've already mentioned that the risk genes are quite common. So up to 24% or so of the population in this part of the country can carry one or more of these susceptibility genes, conferring risk. About 5% of the population around here actually confers a combination of these genes that's very high risk. So again, 5% is quite a large number. And when we go to our step number 2, the immune activation, and ask how often does that occur in the population, I've already told you we can measure that in terms of the autoantibodies in blood. The answer is about 1.6%. So you can see that there's a bit of a barrier here. And it's a bad luck combination to both have the genes and have the activation events and that get you into this 1.6% of the population. But even then, only about 1 out of 4 progress to disease because the incidence of diabetes is about 0.4%, meaning that for every 4 people that have made it to step 2 here, only 1 actually progresses to diabetes. The other 3 stop at this stage, and we'll talk about that in just a few minutes because that's a very important clue for therapy, for knowing what kinds of therapies to try to develop to prevent disease progression. Now, one of the mantras of research is if you can't measure it, you can't study it, right? Some of you have heard that before. So how do you actually study this progression of disease during the phase when the immune system is activated and the immune system is damaging the islet cells? You need to measure the immune system. This cartoon is once again our immune lymphocyte that we talked about earlier with the specific molecules on its surface that are capable of recognizing and killing cells that make insulin. Can we measure those? And here's a prediction of the way we think about disease, which is that during the progression, the number and activation state of these cells should change, probably increasing as we go along. If we could accurately measure that, it would give us a way to monitor not just where is a patient in this progression, but it would give us an ability to monitor whether a therapy that we introduce is actually working the way we want it to work. Okay? So that's being able to measure those cells is a bit of a challenge because in every cc of blood, just pretend this drop on this slide is a cc, a milliliter, a cubic centimeter of blood. There are about 1 million of these immune lymphocytes in that cc of blood. So 1 million of those little black dots that we saw under the microscope early on, okay? But only about 5 to 10 of them have the specificity to attack the beta cell, the cell that makes insulin. So only about 5 to 10 out of those 1 million have the receptor complex on the surface that targets them to the insulin-producing cell. And the challenge is, how can we find them? It's a needle in a haystack kind of a problem to find these cells. And the answer to that dilemma turned out to be to go back to the immune cell itself, take a look at this receptor system that was used to activate it, and mimic that in the laboratory. Use the body's own molecular basis for specificity as a tool to be able to find the immune cells. So I've highlighted here the HLA molecule that I mentioned before, and its co-receptor over here on the immune cell. And when we mimic that in the laboratory, this is the schematic of the HLA molecule. It actually looks like this. It doesn't, of course, look like a green arrow. That was purely schematic. This is what the complex actually looks like. And to make it into a usable tool in research and in the clinic, we multimerize it. We take 4 of these identical little components, link them together around a fluorescent compound in the laboratory, call it a tetramer because of the 4 identical arms, name it after the gene that encodes this part of the molecule, so it's called an HLA tetramer. And this is a probe then that's a mimic for what the immune lymphocyte sees ordinarily in initiating and activating in type 1 diabetes. So I told you this was a needle in a haystack kind of a problem to find the 10 or so out of a million cells in our blood. And basically, this is a solution to that. I like to say it's not hard to find a needle in a haystack if the needle is made out of ferrous metal and you've got a really good magnet, right? So essentially, this is a good magnet. It goes and it finds the immune cells based on their original specificity. And this is what it actually looks like in the laboratory. I'm showing you blood from a patient where the vertical axis means that the cells have bound this tetramer. This, I think I mentioned, this was a fluorescent compound that we engineer into the tetramer so that the vertical axis here measures fluorescence. Each dot in these profiles, which are generated on a flow cytometer, each dot is a cell. So each one of these little red dots up in the upper right panel is a cell that has bound the tetramer. And this happens to be blood from a patient who's had diabetes. He's then had a transplant, a pancreas transplant, to try to cure his diabetes. And the immune response came roaring back and wiped out his insulin-producing cells a second time. And this is what was in his blood during that second wave of attack. So now, is this useful, or how do we propose to help our understanding of diabetes and our ability to intervene? Well, you remember this diagram, which illustrates the disease progression, the history of the disease. And I want to focus your attention on the downward slope over here, where there's progressive loss of islet cells. Because if we can measure the immune response at that point, what our objective is, what we really want to do, is be able to regulate the immune response so that it stops killing the islet cells, stops killing the cells that make insulin, in which case the curve would look like this. We wouldn't have cured anybody because we can't restore the islets unless we do some kind of regenerative process or transplant, or in the future some kind of stem cell process. Right now, what we can do is stop the immune destruction so that you have some remaining islets left. And if you remember, I already told you that we're born with more than we need. So if we can save 50% or so of those islets, that's good enough. Patients aren't patients. They have normal insulin release and normal blood glucose. Now, before I tell you about what we're doing in that regard, I'm going to give you just a little historical vignette here to try and put this in context. As many of you know, before the 1920s, type 1 diabetes, juvenile diabetes, was uniformly fatal because insulin wasn't discovered until the 1920s. Okay? Then from the 1920s until the 1980s, there was a long series of medical progress treating diabetes as a metabolic disorder of high blood sugar, with improvements in the way that insulin was given and the improvements in people's diet. But it wasn't until the 1980s when we realized that looking at diabetes as a metabolic disorder of glucose, of blood sugar, was completely missing the point that diabetes was actually an immune disorder, an autoimmune disorder that we've been talking about, where the immune system kills the cells that make insulin. So the realization that it was an autoimmune disorder came in the early 1980s. It was helped along by our discovery that the HLA-DQ genes, these HLA molecules that I told you about, were the fundamental susceptibility risk factors. That's when the autoantibody testing was first introduced. That's when we and others first cloned the lymphocytes from blood that are causing this disease. And that's also when researchers started to develop mouse models of type 1 diabetes to study it better. Didn't take long. The 1990s developed somewhere on the order of 40 or 50 different ways to cure and reverse type 1 diabetes in those mice based on changing the immune system. Okay. First clinical trials were attempted in patients, and at that point, they used major drugs that interfered with all immune responses. These were not good therapies long term, but they were enough to prove the point that interfering with immune activation was beneficial in terms of protecting patients from progressive loss of islet cells. The standardization was done for autoantibodies. That's when we started these large national programs I already mentioned, now over 200,000 people screened. That's also when we developed laboratory assays to test some of the new drugs. And there were some major advances in terms of purifying islet cells used for transplantation. And now here we are in the first decade of the 21st century. And we're in this phase where we're very actively performing clinical trials in patients with diabetes and in their siblings and children who are at risk of diabetes to try to block and modulate the immune response. The predictive testing that I've already referred to involving genetics and autoantibodies is very standardized now. It's worldwide. It's currently still run in academic centers, but ultimately will be a general medical test. And then the prevention trials, true prevention trials in people who don't have disease yet have been started. So I'd like to talk about this as a progression, a decade of discovery followed by a decade of innovation, followed by the current decade that we're in now, the decade of clinical trials. And this is for real. This is the current list. How many is it here? 17? No, so 14. I've got 7 in each column. 14 different clinical trials that are either currently going on or will start later this year. All for intervening in the immune response to modulate the progressive destruction of the beta cells. So we're right in the midst of this major thrust to take the laboratory findings of the immune destruction of the islet cells and make it into something clinically important for clinical trials. And what do all those trials do? Well, the vast majority of that list that I just showed you on the previous panel, the vast majority of those are compounds which interfere with one of these points, which I think hopefully you now are all familiar with. These are the points of intersection at which the immune cell learns its specificity. It needs to engage these molecules to be activated. And so selective targeting with new therapeutics at each of these 4 points have been developed and are currently in clinical trials. The trials actually are organized through the National Institute of Health. They're run nationwide. This is a diagram of all the participating centers. There's a 800 number. There's an email. Anybody who's actually interested in learning more about this, very easy to do. If you don't have a pencil with you and you're watching this at home, It's very easy to remember, diabetes@benaroyaresearch.org. And you get directed to the trial center closest to where you live. The Benaroya Research Institute is the trial center for the 5-state region in the Pacific Northwest. So anybody in the Northwest will get referred in here to Seattle. Now, this is still research. And the reason it's still research is because we don't know yet if any of those therapies are going to work well enough to answer these 3 key questions. Okay? Are they safe? Well, I've already told you that the immune response not only attacks tissue and causes diabetes, but it protects us from infections. And so we have to go through the clinical trials experience with all of these trials to find out if we are inadvertently causing risk of infection to any of the people taking these therapies. So this is a very fundamental issue, very fundamental question. It's why trying to develop these therapies that are very specific for the surface molecules that target the islet cells is an attractive therapy direction. It should be a better way to develop therapy compared to therapies that ablate the immune response more generally or more globally. Second question I've put up here is, do you need to take these drugs for a long time? Because the ideal therapeutic would redirect the immune system to mature in a way that restores the way that it was before the immune attack. And therefore, it could potentially be a short-term therapy. So some of those trials that were on that list, of those 14 trials, some of them are therapeutics that are taken just for a short period of time, a couple of weeks in the shortest case. Some of them are therapeutics that need to be taken for a very long period of time. We don't know yet which are going to be the best and which are going to work. And the third question I put up here is sort of a challenge to all of us in the research world, is we know so much now about the immune attack and about the immune destruction. Why can't we figure out how to restore the normal immune balance? And so that's the topic I'm going to talk about for the remainder of today's discussion, is how are we trying to figure out how to restore the normal immune balance? And so I'm going to start with a simplified cartoon of a normal immune response. Okay? So just picture this little curve as the magnitude of an immune response. And it can be an immune response against anything. A cold virus. Okay? What does your immune system do, for example? It doesn't have to be a response against cells that make insulin. It can be a response to any challenge. Well, the immune response first responds with it ramps up to a very great degree. It expands. It's a process of cell proliferation, cell activation, secretion of molecules that are called cytokines. A lot goes on in this early activation phase of an immune response. But it doesn't last forever. In the case of an infection, the immune response can clear the infection, and then the immune response starts to fade away because it's not needed anymore. So this downward part of the curve is the fade away of the immune response, the start of the resolution phase. And eventually the immune response comes down to sort of close to what it started at. It has other features at this point that are called immune memory, but fundamentally it has resolved most of the acute parts of the response. So that's what's stated here. The immune response is a balance between this kind of active inflammatory phase and this kind of resolution phase down here. Now, what we now know is that this resolution phase, this downward phase, is not just a passive going away or dying off of the immune response. It's actually an active process where the immune response turns itself off. And one of those mechanisms of turning itself off is through a type of immune cell called a regulatory cell, whose purpose is to tone down the immune response when the immune response is no longer needed. So that's cartooned here as well, of regulatory cells that can turn off these aggressive immune cells. And here's what it looks like in the research laboratory when this happens. These are, again, profiles on a flow cytometer where every dot is a cell. Once again, it's an experiment using those tetramer molecules I told you about to be able to look at the immune cells. The middle panel here shows you the cells that are binding those tetramers. In other words, these are the cells that are causing the damage to the islet cell in a patient. They're circulating in the blood, and we've detected them here using the flow technology. And in the laboratory, when we add these regulatory cells from the patient, the same patient that are educated in the laboratory to be regulatory, we can markedly diminish these cells, almost back to normal. And so the good news is we think this is doable, that the immune system can learn to regulate itself. The bad news is this is all in the laboratory. This is not in clinical trials yet, although we hope it will be soon. But this is basically a proof of concept laboratory step to get us to where we want to be for additional clinical trials. But that's an illustration of the type of outcome that I was referring to in terms of trying to develop therapeutic strategies that are based on restoring normal immune balance. I think I'll end there and take questions from the audience. I want to be sure and acknowledge a number of collaborators who have been instrumental in this program and in the work that I summarized with you today. The individuals on the left of this are our clinical collaborators. Carla Greenbaum is the coordinator, the head of the coordinating center for the TrialNet, that large NIH-sponsored consortium network that I already told you about. The people on the right here are a number of laboratory investigators who at various points have been instrumental in the work I described. And I also want to acknowledge both the Juvenile Diabetes Research Foundation and the National Institute of Health for funding support for these studies and this kind of research program. So thank you very much, and I'll be happy to take questions.
You said that in the Northwest, there were 24% of people who were at risk. Does this number look different if you look in different parts of the country, and what determines that frequency?
Right. That's a good question. There's a lot of variation in our genes. Different, of course, the genes that any individual has depend on what genes their parents had. But when looking at large populations, there's major genetic variation among the major ethnic groups that exist. So, for instance, the type of diabetes susceptibility genes that cause autoimmune diabetes, type 1 diabetes, are much more common in Caucasoid-type populations than they are in Oriental-type populations. So it's not surprising that the incidence of autoimmune diabetes in Japan, for example, is a fraction of what the incidence is here in the United States. That's all because of the baseline frequency of these genes is about 5-fold less in Japan, for example, than it is here. So there's a lot of ethnic stratification that can go on that can help in terms of selecting the right populations for the screening studies that I described.
So a related question perhaps, you mentioned that about 1.4% of people move to this activation stage, but only a few of them end up getting type 1 diabetes. Right. For the rest, if they live long enough, will they get type 1 diabetes, or are they somehow stopped in perpetuity? Right,
that's also an excellent question. So the 3 out of 4 individuals who have gone through the activation stage but have not yet progressed to disease, what is it about them that keeps them from progressing? And the answer seems to be that they have an immune response that has learned to counter-regulate itself. The exact reason for that has yet to be discovered. That's an area, very much an area of ongoing research. Most of the current work suggests that it has to do with these regulatory cells that I mentioned at the very end of this talk. We want therapeutically to figure out how to use those regulatory cells to keep people from progressing to clinical disease. It may well be that the 3 out of 4 individuals who have figured it out on their own naturally to stop progressing have done so because their regulatory cells have kicked in. They've either been boosted by something environmental, which is one of the areas being researched right now, or they have other genes that are so-called protective in the sense that they help the activation of those regulatory genes. There are, of those regulatory cells, excuse me. So there are a number of genetic modifiers that have been identified in the last couple of years that are capable of doing that. And whether that turns out to be the answer, that having or activating those particular genes leads to activation and expansion of the regulatory response, leads to stopping the progression to disease, whether that's the actual pathway or not, is very much why we still need a little bit of research in this area. But that's the dominant hypothesis right now.
In your opinion, is it possible to solely implicate the thymus in autoimmunity, the organ from which these cells are matured and developed? I was curious if the test for avidity that these cells have to undergo is somehow skewed in a way because of miscommunication between the thymus or whatever, and if that's possible, if it's perhaps an issue of genetic mutation or whatever.
Right, that's also a great question. The background to the question, for those of you who aren't familiar with the thymus, is that the thymus is an organ that sits here, and one of its functions, starting neonatally actually, but one of its functions early on in life is to be the place where your immune cells mature, where they develop, where they first learn how to expand, and they acquire these molecules on the surface that are the same molecules I was pointing to earlier that give those cells their specificity for what they're going to respond to in the future. Once they leave the thymus, they go out into your blood, they circulate in the body, and they are your immune response. Now, so the question is that where the problem lies? Can we blame it all on the way the cells develop in the thymus and acquire those particular molecules? And I think the answer is probably that the first step there, the susceptibility step, which is, as I say, it's blamed on your genes. What those genes do is they make the molecules that sit on the surface of these cells and give them their specificity. That is, in a functional sense, the determinant of which cells are going to escape the thymus, which cells are going to be able to leave the thymus, get out into the peripheral circulation, and ultimately be the cells that get triggered, that get activated, and that cause the damage and destruction. We know from experimental models in mice that if you figure out how to stop those cells from leaving the thymus in the first place, you actually protect from the autoimmune disease. So at first blush, the answer to the question is yes, that's where the problem lies. If the autoimmune cells didn't get out of the thymus, they wouldn't be able to cause the disease. We also know from mice that mutations, just like the question asked, mutations in the thymus can cause more cells to leave than usual. That gives mice an earlier and more aggressive autoimmune diabetes. So the answer is, it looks like mutations in the thymus and barriers to thymic development are actually reasonable ways to modulate whether the cells in the periphery are there to cause autoimmune disease. Okay? So that's one part of the answer, but that's not all of it, because the ability of these cells to mature in the thymus, of the immune cells, to mature in the thymus and get out into the periphery is not just a matter of causing diabetes or not causing diabetes. It's actually a matter of being able to provide cells in the peripheral circulation that can protect us from other things too, the normal immune functions, like protecting us from viruses and bacteria and the like. And so we can't just go in and block the maturation of cells in the thymus, because that would lead to immunodeficiencies. And that's not a viable form of therapeutic. In addition, the exact nature of the molecules that are on the immune cell surface have some subtleties to them that make it very difficult to selectively block the release of the autoreactive cell, the immune cell that causes diabetes, without also blocking the release of some of these other cells that we need to protect us from infection. So right now, none of the therapies that I mentioned that we're developing are targeted at the thymus. All of the therapies instead are targeted at either getting rid of the autoreactive cell, the immune cells that are in the blood that cause the disease, or are targeted at boosting the regulatory cells out there in the blood that we think can modulate and counter-regulate to help in the disease. So it was a long answer to a complicated question. Yeah.
So I have type 1 diabetes, and I have a daughter who has type 1 diabetes, and my newest daughter is in the TEDdy study because she shows the marker for developing type 1 diabetes. And my question is, since it is an autoimmune disease, well, basically, I'm hoping that in doing doing the baby's immunizations, that if I don't do them all at once, you go in for a visit and you have, the baby has 4 shots, and I'm wondering if that triggers such an autoimmune response or such a immune response that it could, she could end up.
Right.
Her body could end up killing the wrong cells. And I'm wondering if I space those out a little bit more, which we're doing, is that helping at all or in preventing the diabetes?
Yeah, another great question. First of all, let me commend you and thank you for participating in the research studies. That's a very altruistic and remarkable thing that people in the community need to be part of the team to help figure this out. So thank you and your family. With respect to what are the triggers that take somebody who is susceptible and move them along in this cascade, and to what extent vaccines are the triggers, and to what extent infections are the triggers. This is not an easy answer because the immune activation, just as you suggest, the level of immune activation is very likely to be one of the modifying factors that is part of that threshold, the part of taking the 5% high-risk group and moving to the 1.6% antibody-positive group and to the 0.4% disease group. So it's a trade-off of being at risk of the infections. We certainly don't want high-risk kids to be getting the mumps and things like that, because that is one of those things that might be part of pushing the cascade along. But there is also a theoretical risk, just exactly as you outlined, that an overstimulation of the immune system, perhaps through combinations of vaccines, might also be a potential risk factor. So what you're doing in terms of spacing out the vaccines is, to my way of thinking, a very prudent thing to do. You protect against the infection, you try to minimize an unknown risk that might exist from too much stimulation. There is no good, hard data to answer your question. So it really is a matter of opinion, and I just gave you mine.
Okay, great.